All right, today we're going to put on our detective hats and dive deep into the microscopic world of infectious disease. We're going to find out just how scientists crack the case. You know that feeling, right? You're just down for the count. Fever, cough, feeling absolutely awful. But the real question is, what's actually causing it? Who's the invisible culprit here? Well, to get that answer, microbiologists basically become detectives. They've got to gather clues, analyze all the evidence, and build a solid case against a very specific microscopic suspect. So let's follow along and see how they do it. So, our investigation kicks off inside the human body, but this is not your typical crime scene. It's an invisible one, and it is absolutely teeming with countless potential suspects. Okay, so right off the bat, our microbe detectives hit a huge challenge. See, your body is home to literally trillions of microbes. Most of them are totally harmless, some are even helpful. That's what we call our normal flora. So when they find a potential pathogen, the big question is this thing the actual cause of the sickness, or is it just an innocent bystander that happened to be at the scene of the crime? To solve any case, a detective needs to know who they're looking for. And in microbiology, that means you have to profile the main types of microbes that can cause all this trouble. So, in the world of medical microbiology, you've got your usual suspects, and they generally fall into three big groups. First up, you have bacteria. These are prokaryotic cells with a cell wall but no nucleus. Then you've got viruses, which honestly are more like little nucleic acid delivery machines, just a piece of DNA or RNA wrapped in a protein shell ready to hijack your cells. And finally, we have fungi, which are more complex, things like yeasts and molds. Each one has a totally unique signature. Okay, let's zoom in on one of the most common groups of culprits, bacteria. Just like you'd profile a criminal, scientists have to build a really detailed profile to pin down exactly who they're dealing with. So, how do you build that profile? Well, it's kind of like a detective's ID checklist. They go down a list of key questions. How does its cell wall react to certain stains? What's its basic shape and how is it arranged? Does it need oxygen to live? And what does it like to eat? Its biochemical properties. Every single answer is a clue that helps narrow down the suspect's identity. One of the first things they look for is shape. It's super basic but super important. For example, if the bacteria they see under the microscope are little spheres, they're classified as cocci. And on the flip side, if they look like little rods, they're called bacilli. Just being able to tell the difference between these two shapes, cocci and bacilli, is a huge first step in the whole identification game. Now this is where it gets really cool, the Gram stain. Based on what their cell wall is like, bacteria will stain one of two colors. This one simple test splits pretty much all bacteria into two giant groups, Gram-positive and Gram-negative. And this is why it matters so much. This simple color difference is a massive clue for doctors. It helps them make a really educated guess about which antibiotics might actually work, guiding treatment way before they have a final ID. All right, so we've got a profile of our suspect. Now it's time to break out the high-tech forensic toolkit and get a direct look at the evidence. And just like in any investigation, you've got to have the right tool for the job. A standard light microscope? It's great for most bacteria and fungi. For some really tiny bacteria, you might need a special dark field microscope. But viruses? Oh, they're a whole different ballgame. They are so incredibly tiny, you need the massive power of an electron microscope to even see them. And of course, all those